So when he crossed those F1s, he got what he expected. He got some kids that had white flowered plants. He had seen organisms do this, right? Two brown-haired parents have a blonde-haired child, so he wasn't surprised by this. But where he separated himself from other people who tried to figure this out, people like Charles Darwin, for instance, he counted, and he was a math major. So the numbers meant something to them. When he did this with pea plants, looking at each of the 7 traits that he chose to look at, separately looking at each of the 7 traits, he always got a ratio in this F2 generation, the F1's kids, of 3 dominant phenotype individuals for every 1 recessive phenotype individual. And this 3-to-1 meant something to him. What it told him was this. He said, I know what would give you 3-to-1 if you do what I did. If you took 2 purebred parentals, made F1 kids out of them, and crossed the F1s to make an F2 generation, I know what would give you 3-to-1 ratio, dominant to recessive in the F2s. That's if each of these individuals, parents, offspring, whatever you want to call them at the time, if each of these individuals had 2 things that cause flower color, 2, but they gave their kids only 1. So if each of these pea plants, for instance, had 2 flower colored genes, I'm going to call them genes now, and the purple flowered parental parents, the pure ones, the true breeders, had both of those 2, the purple flower color, capital P. That means we're going to mate them. The X's means we're going to cross them. That means one's going to be mom, one's going to be dad. And the white flowered pure ones had only white flowered stuff. We use lowercase p to mean recessive, just like he did. We keep that convention. If this is how it works, when these 2 parents have kids, they only give 1 of what they have 2 of to their kids. That means all these F1 kids would get a dominant purple flower gene from this parent, a recessive a white flower gene from that parent, and since purple's dominant over white, they would be all white flowered. That would explain that. And then when these kids became parents, when you cross the F1s, if they gave them only 1 of what they had 2 of, he said, then this is how it'll work. This parent over here will give kids either the purple flower gene or the white flower gene, but it'll only give 1 of them, which we emphasized or symbolized by separating the 2 letters as I drew them now. And if this parent, when they had kids, they gave only 1 of the flower color genes they had, either the purple one or the white one, then these are the kind of kids I get. You notice I'm drawing a Punnett square that you're used to, right? Right? If each parent gives the purple flower gene, then you'll have a kid that has both purple flower genes. If one parent gives the purple flower and one parent gives the white flower, which can happen 2 different ways, right? Purple from mom, white from dad, purple from dad, white from mom. You get 2 of those. And if both parents give the white flower gene to their kids, you get 1 of these. Well, all 3 of these show purple flowers. 3 purple, dominant to every 1 right, recessive. He said, if this is how heredity works, everybody has a pair of factors for each trait, like flower color, but they only give 1 of that pair to their kids, then this is what I'll get if I do these 2 crosses. Cross the parentals, then cross the F1s. This is what I'll get in the F2 generation. 3 dominant to every 1 recessive. So he said, this is how I think it works, because this is what I get. And all that thinking is what he called there's his actual numbers. Those are his actual numbers. You see how many crosses he did for each of those 7 traits, getting that 3-to-1 ratio. All those thoughts he called his law of segregation. Segregate is a word that means separate. He chose that word. for this idea. He said, I think every organism has two factors. These again are now what we're calling genes, but we were 100 years away from that word when Mendel was doing these crosses. Where'd they get their two from? One from their mom, one from their dad. What are they going to give their kids? Just one of what they have two of. Because when gametes form, and gametes were known to exist by this time, egg cells and sperm cells. But what was in them, genes and stuff, that was 100 years away, right? But he said, whatever this stuff is, when parents take the first step to becoming parents, which is make gametes, everything they have pairs of separates, segregates, and the gametes, and therefore the kids, get the keywords in this whole thing. One from each pair. One from each pair. That is, in a nutshell, the law of segregation. So, this I can't emphasize enough. It's the key to this whole unit. It's the key to a couple other units. It's the key idea to meiosis, which was undiscovered at Mendel's time. Nobody had yet looked under the microscope, watched cells undergo meiosis, and described all the steps and the two divisions and the stuff we've already talked about. So this is what put him way ahead of our time. He was correctly describing what happened with chromosomes and the genes that they are made of in meiosis before anybody had even discovered meiosis. Okay, so there. This, I'll say it again, right? But now we'll add this. What events of meiosis account for the separation of pairs so that gametes get one from each pair? It's the separation of the DNA copies produced in S phase by replication. It's the separation of these DNA copies. But before the copies get separated, the homologous pairs get separated. So that by the time, I've kind of made a mess of this, but by the time replication happens and two homologous single-stranded chromosomes are now double-stranded, copies made, and after the first meiosis division, this chromosome separated from that one, and after the second one, the chromatids are separated so that we have four gametes that each have one of what this parent cell had two of. The events in meiosis that account for this are the lining up of homologs in meiosis 1, their separation in anaphase 1, and then the separation of chromatids in anaphase 2. So, this is how Mendel figured out that very key basic idea of pairs and one from each pair. Okay, so now we have these words, right? Now that we've identified and called these things genes, we use the same kind of symbols that Mendel used, alphabet letters. This is where we have to be careful. We now know what we're talking about is DNA. This stuff is DNA. And we used alphabet letters to symbolize those bases, right? A, T, C, and G. Now we're using alphabet letters to symbolize something different, and sometimes we even use the same letters as we did before, like T's and A's. So now if we, over here, draw what represents a chromosome, right, which is one long strand of DNA, and if we say, okay, this part right here, that's the part that might unzip, be used to make some RNA, that's used to, that results in a protein that causes blue eyes. Now we're saying we're going to call this little stretch of DNA a blue-eyed gene, and we're going to use an alphabet letter to represent it. So that's what these represent. So back to Mendel's crosses, his parental generation. Each parent had two of the same kind of genes, 4 traits like flower color or whatever. Now we call this pure, we call this true breeding. Now we're going to call it homozygous. Same, homo meaning same in this case. The two genes they have for that trait are the same. Big T, or little t, whatever, right? Or if they're different, then heterozygous. So all the F1 kids in Mendel's crosses were heterozygous. They got one dominant gene from one parent, one recessive gene from the other. Okay, so we've already said what phenotype and genotype is, right? So now what we're saying is that to describe something's genotype, we can either use words like heterozygous, that tells us it's got one dominant gene, one recessive, or we can use alphabet letters, right? We can say that's its genotype. Homozygous dominant, we can say it like that.